Today we have Dr. Juan Dominguez, Dr. Fahd Qadir, and Dr. Mayur Doke, as well as Isabella Altilio, who is a graduate student in the laboratory. And the title of this talk is Pancreatic Regeneration at Work, Lessons from Dynamic Single-Cell RNA-seq of Live Human Pancreas Slices. And just a short bio on each of our participants. Dr. Dominguez is the director of Stem Cell and Pancreatic Regeneration Laboratory and the research professor of surgery and cell biology at the DRI Miami, University of Miami Miller School of Medicine. Before joining the DRI faculty, he worked at the Roslin Institute, actually in Scotland, known for the cloning of Dolly the sheep, and he obtained his PhD there and acquired considerable experience in embryonic stem cells and state-of-the-art genetic engineering techniques. Working in collaboration with other DRI faculty and collaborators, he leads several projects that is focused on harnessing the natural ability of the human pancreas to regenerate its endocrine compartment, as well as stem cell-based approaches to restore beta cell mass in context of type 1 and type 2 diabetes. Sorry, just letting people in. The group has a cell metabolism short video that kind of, details their paper for World Diabetes Day, which I will share in the chat. Bunch more people joining. Dr. Fahd Qadir is currently a postdoctoral fellow at Tulane University School of Medicine in the lab of Dr. Frank Moves Jarvis, and he's interested in studying the role of sex hormones and chromosomes on islet biology and function. Prior to joining Tulane, he graduated from the University of Miami Molecular Cell and Developmental Biology graduate program at the DRI, where he studied long-term culture model of live human pancreatic slices, enabling the study of islet biology and regeneration over extended periods of time. The current paper in cell metabolism, again, which contains work performed largely by Dr. Mayur Doke, extends upon the prior model, establishing a dynamic longitudinal system using cultured human pancreatic slices. And this work will allow for longitudinal study of pancreatic slices to work as a novel model system in beta cell regenerative therapy and type 1 diabetes research. Again, we will put that link to that paper in the chat. Dr. Mayur Doke is a postdoctoral researcher in Dr. Dominguez beta cell regeneration lab at DRI Miami, where groundbreaking work is being conducted on regenerative strategies for type 1 diabetes. Specializing in stem cell differentiation and islet regeneration, Dr. Doke's focus lies in the induction of human pancreatic ductal BMP responsive progenitor cells and using cutting-edge human pancreatic slice HPS systems and single-cell-seq data analysis. He successfully traced the true cell fate trajectories of progenitor ductal cells as they differentiate into beta-like insulin-producing cells through bioinformatics analysis. Cannot wait to hear more about that. And finally, last but not least, Isabella Altilio is a PhD candidate at University of Miami. Her long-term goal is to understand the mechanism of action behind metabolic diseases and associated comorbidities. And at University of Miami, she joined the Dominguez lab and is actively performing studies related to diabetes and regeneration of the pancreas. Most importantly, she's led the project in a direction of clinical trials to pursue a cure for type 1 diabetes. Welcome. Thank you for joining us. This team is coming from across the globe, really. We've, Isabella's coming to us from South America, and we also have Dr. Doke and Dr. Dominguez from Miami, and finally, I believe Dr. Qadir is down at Tulane. So it's great to have everyone here, and thank you for coordinating your schedules and making this happen across several time zones.
Thank you, Monica. Thank you very much. So I'm going to share my screen, and then as we have people from different places of the world, they will jump in and we'll manage to make this as seamless as possible. All right. So
fantastic.
Start sharing my screen here.
Feel free, guests can feel free to drop questions in the chat during the talk as well.
All right. So if you all see my screen, today we're going to discuss our most recent work on pancreatic beta cell regeneration. And this is work that was co-led by my colleague, Ricardo Pastori, and myself here at the University of Miami. And from here, we are going to discuss part of the paper, Mayur and I, and then, Fahad and Isabella will pitch in different, different parts of the presentation. discuss what they did.
Great.
I was charged with giving a brief introduction, and I'm sorry this is very general, but it's important to know the basics when we explain what the ducts do and everything. So the pancreas is rather unique in the GI tract because it has two very distinct functions. And as you very well know, most of the pancreas is actually exocrine. It makes digestive juices, and the cells that do that are the acinar cells. Those digestive juices are collected through a vast network of pancreatic ducts, and those take the pancreatic juices to the duodenum. And then, of course, the area that we like the most, because it has a direct impact on people with type 1 diabetes, is the islets. The islets of Langerhans are only 1 or 2% of the organ, but they have cells that make insulin, glucagon, and other hormones that keep sugar levels within a normal range. So here we can see in the next slide how it looks like when you look at the microscope, the pancreas. You have a healthy islet in there, surrounded by all the acinar cells around it. And when you have type 1 diabetes, there's an autoimmune attack. So autoreactive T lymphocytes infiltrate the islet and destroy the beta cells that make insulin. And as a result, people with this condition need to take insulin for life. So obviously there's a lot of interest in replenishing those beta cells. And when you have type 1 diabetes, there's cell replacement strategies. Islet transplantation has been a major focus of our institute for the past 30 years or so. Now there's stem cell transplantation as well. But my group and others around the world are also interested in inducing endogenous regeneration, which is to harness the ability of the pancreas to heal itself and regrow new islets. So the idea that the pancreas can regenerate itself and that islets can regrow in the adult pancreas comes back more than 100 years. That's Robert Bensley at the University of Chicago, who in 1911 posited that islets do in fact come and arise from all the ducts. So you have he described in excruciating detail and have these beautiful drawings of islets that are obviously sprouting and connected intimately with the ducts, right? And he claimed that that was the origin of islets. Now, this is still a little bit controversial because of, we don't have the time to go into it, but some papers on transgenic animals seem to suggest that this was not something that was happening in the adult pancreas. But there's no dispute whatsoever that this is the way that the pancreas forms. During normal embryonic development, new islets sprout from ducts, and this process is called branching tubulogenesis. As ducts grow and sprout, they create new acinar and endocrine tissue. Right, so that's what happens during embryonic development. What is true, and this is more or less a consensus now, is that in the adult healthy pancreas, not much happens. And beta cells are extremely long-lived. In humans, they can last decades. And if something happens to one of them, normally replication of another will take care of the problem. So that's probably what the mouse models in most of the mouse models have been looking at. And indeed, there's no regeneration unless there's a need for it. But now things start to get interesting when there's a stress in the organ, and that stress can be metabolic or immune. And then weird things happen. For instance, beta cells de-differentiate. They revert to a progenitor-like stage, and then they can redifferentiate into more beta cells or even other endocrine cell types. You can also see how alpha cells can transdifferentiate or become beta cells. And those things happen when you have stress in the organ. And what we and others contend is that when you have extensive damage to the pancreas, the likes of autoimmunity or pancreatitis, in those situations, you have a partial reactivation of the embryonic program that led to the formation of islets from ducts. So that's actually what I'm reminded of when I look at this picture, and I present it in my every time there's a communication, I like to put it. This is the Eagle Nebula, some 7,000 light years away from Earth. And that region concretely in particular, that's called the Pillars of Creation. It's a vast cloud of gas, interstellar gas, that is condensing and forming new stars all the time. And when I look at the pancreas of many type 1D donors that we get from MPOL, I see something similar. I see clouds of ductal tissue in green and new islets that are completely intermingled with them. And in other cases, like in here, you can see lots of small islets or stars, if you will, that are forming, coalescing from the tip of the ducts. And clearly, when you see this pancreas, something is happening there. There's a compensatory response, there's ductal remodeling, and what we think is happening is that this is a nursery. The ductal system is a nursery for new islets. Obviously, we know how this story ends because autoimmunity eventually gets, the last word, and you have diabetes. But the pancreas is doing its best to regenerate, and this process is long lasting. You have sometimes patients in their 90s who have been diabetic for 50 years or longer, and when you look at their pancreas after they pass away, they still have signs of beta cell regeneration. So the pancreas is trying actively to correct this problem.
Quick question there. I don't know if you guys can answer this, but when the when once someone is diagnosed, a child is diagnosed with type 1 diabetes, just say, let's pick that time frame. After insulin therapy has begun, there is the honeymoon period begins. And I wonder if anyone has noted, or if your lab has noted, this kind of phenotype happening during that sort of honeymoon recovery of function.
Yes, that's an active area of study, and obviously we don't get access to many of those samples, but indirectly, clearly there's a recovery of beta cell function. In many instances, it's due simply to the fact that after onset, the patient is immediately put on insulin, and that kinds of gives a rest to the islets, right? So they immediately improve their function. But there's a lot of studies, and we are collaborating, in fact, with a group in Spain. They are looking at microRNAs involved in what they call partial remission during the honeymoon phase. And many of these microRNAs are involved not only in immune modulation, but also in regeneration. This is what happens, what we think is happening. Indeed, when you are started to correct hyperglycemia with insulin, that allows for the pancreas to recover a little bit. And there may be a lot of regeneration happening at that time. But that in itself is like fuel, if you will, for the immune system. So it comes back with a vengeance, right? And that's what happens eventually. But
there's some evidence of sort of ductal transitioning into islet or beta cell phenotype during that time frame, or no?
There's no direct evidence, simply because it's very difficult. I mean, many of the cases that we get, I would have to check, but you can have many of these sample cases that we look at are early onset diabetes, and they may or may not be in the honeymoon phase. Quite frankly, the clinical history of these donors is not complete at times, but I wouldn't be surprised. I mean, this patient, in this donor in particular, he was or she, I cannot say, it's a pediatric donor, and he or she may have been in the honeymoon phase. I don't know that, but it wouldn't be surprising, because again,
it's an interesting window of time.
And there's a lot of interest in prolonging this as much as you can, because if there's active regeneration and perhaps the immune system is a little bit subdued after this onset, it would be great to postpone the onset of diabetes, or to prolong the length of this honeymoon phase for as long as you can.
Okay, thanks for that.
Right. So a number of years ago, we hypothesized that if there are progenitor cells in the exocrine pancreas, and particularly in the ducts, they should respond to BMP signaling. Why BMP signaling? BMPs are, growth factors that are circulating in your bloodstream and in mine, everywhere. They are like fuel for stem cells across many organ systems. And BMP7 in particular was of great interest to us, because it has been used clinically already. It's used all the time to grow bone when you do spinal fusion, spinal surgery, and you have to fuse two vertebrae. You use a glue that is made of BMP7. So there's already, it has clinical credentials, and it would be easier for us to take to the clinic. What we noticed is that when we take the exocrine tissue of the human pancreas after you do an islet isolation, and people in the cGMP facility were throwing away the rest of the pancreas, we told them, save it. Let's see what happens when you have BMP7. What we noticed is that islets started to grow, and I don't say islet lightly, because many people see something like this and claim, oh, look, it's an islet. No, these were actual collections of endocrine cells, of all the endocrine cell types, and they produce insulin in response to glucose stimulation, and even corrected hyperglycemia when transplanted in diabetic animals. So islets form when you add BMP7. So after this, we published a number of papers over the last few years characterizing the cells that responded to BMP7. And they all happen to be located in the lining of the pancreatic ducts. And they were kind of hiding in plain sight. The cells that we want are the red ones. They express ALK3. That's the receptor to which BMP7 engages. And they also express PDX1, which is a major architect of the pancreas. So there's no surprise in there. And they're intercalated among the green cells, which are the normal ductal cells, but they don't express a key marker that normal ductal cells do. So Fahd actually was key in doing all these experiments that we published back in 2018, where we isolated these cells, separated them, put them in culture, and we showed that when we expose them to BMP7, these progenitor cells expand. And when you remove the BMP7, they differentiate into all the different cell types that you find in the pancreas, including functional beta cells. One of the most beautiful experiments, I think, that we did was one where we isolated these cells and transplanted them into immune deficient mice under the kidney capsule. So these cells were isolated from human donors, and they were transplanted as single cells that didn't express any of the mature markers of any of the lineages of the pancreas. So after a few weeks, when you look at the kidney of that mouse, you had kind of a mini organ, a mini pancreas. You had small islets, you had perfectly formed acini with the acinar tissue. They were seemingly draining into small ducts. So to us, that was mind-blowing, the fact that these cells have the ability not only to differentiate into all these different cell types, but they have the memory to recreate a mini pancreas of sorts. They have the memory to create an organ, if you will. So then we have spent the last few years doing studies on this model, and I'm going to let Fahd continue with that. It's called human pancreatic slices, and as he will explain, this is exactly what it looks like. These are thin sections of the human pancreas that we can keep alive for quite some time. Fahd, go for it. I'm going to stop sharing my presentation, and you can go
All
with
right. All right, so everybody can hear me, right?
Great,
Okay.
perfect.
So this is just a small video. Thank you, Monica, for the introduction in the beginning, and thanks, Juan, for a nice talk so far. So I guess I should start by saying that the pancreatic slices, or the generation of pancreatic slices, is actually not something new. This is something that Stephen Spear from Germany actually has worked a lot on, especially him and Alejandro Caicedo's lab as well. But the issue with these slices is that when we would culture them in excess of 24 hours, they would degrade, largely due to the fact that majority of the pancreatic slice is acinar, as is well known, right? So the digestive juices that were generated from the acinar, when you have them in an environment at 37 degrees in an incubator, they would just digest. So literally the slices would just fall apart. So this is just an image of how we do the slices. I mean, this is pretty normal. I mean, pretty sort of normal and regular for us now. What you're seeing over here is a blade that is spinning very fast, and it's cutting through this block of pancreata, which are like embedded in low melting point alveoli. So this is how we generate the slices. They're about 125 to 135 microns in diameter. So once we get the slice, what you can see over here is basically, the acinar tissue, the ducts, the islets, and what you can appreciate is that we get the complete morphology or cytoarchitecture of all pancreatic cell types. And when we actually go and stain for them, we can actually see, the islets, which you see over here, stained in red, and the rest of the tissue, which is stained in blue, right? So the issue is, as I said, that the minute we put these slices in culture, they're going to degrade very rapidly and just literally sort of melt away, and you see a loss in the overall cross-sectional area and the health of the slice, and they become non-functional. So this was quite a bit of a challenge. So work that we did along with Chris Fricker, who's a islet physiologist, I would say, and what his sort of interest is looking at, oxygen levels and saturation levels of islets. So we started thinking that, well, maybe what's happening is that the slices, owing to the thickness, is the fact that they're really basically choking. So they're not able to be aerated well. So you have a loss of, anaerobic respiration, which is really what is necessary for driving, tissue health. And when we modeled this using computer simulations, and I'm not going to get into this, but what we found was that there were hypoxic zones that would exist in the core of the slice, which is something which would be understandable purely because of the fact that, the slice is very thick. So what we did is that we went to try and address this question, and this is just data from our paper, and you can see over here, for example, that HIF1 alpha, which is hypoxia inducible factor 1 alpha, is a factor that is produced during situations of hypoxic stress. You can see that they actually were reduced in this culture system that we created. Now, what is the uniqueness about this culture system? Well, I'll talk about this, but what Juan actually designed was a dish known as the PFC dish, or we call it the air hive. And what the PFC dish is essentially that it's made of this very thin membrane constructed of perfluorocarbon. And what the uniqueness of that membrane is that it's only permeable to gas, not to liquid. So you could, unlike a transwell where you'd have to put liquid on both sides of the membrane, you could put liquid only on one side of the membrane, and it would allow for an air-liquid interface to be generated, allowing for, double the amount of oxygen essentially to be perfusing through the media and through the tissues. And in addition to this, we had to address the problem of how enzymes would degrade the slice themselves, right? So what we started adding to this was a cocktail of different enzyme inhibitors, and we detail all of this in the paper. So if you're interested in looking at that, I would suggest going and reading the paper in detail. But the unique media conditions alongside the PFC dish resulted in some really interesting results. First of all, we saw that the overall cross-sectional area of the slice, it didn't degrade over 14 days of culture. And then when we took slices at about 10 days and we put them through a dynamic GSIS, or a dynamic glucose-stimulated insulin secretion assay, we found that, the slices were, bihormonal. They would secrete both insulin and glucagon, and, show good first phase and second phase insulin, and, we'd also see glucagon secretion from advanced slices. And, work done by a good friend of mine, John Weitz. What he did was, from when he was a grad student in Alejo's lab, so we were both grad students in Miami at the same time. So he took these slices and then he looked at calcium signaling, and he found islets that after 10 days of culture in PFC dishes, showed good oscillatory response in terms of calcium dynamics. So what we're trying to show over here is that, physiologically, the slices were very healthy. And when we went and we did staining, so this is work that was done by Sylvia, like brilliant lab manager in our lab. So what Sylvia did was that she went and she stained for the slices, looking for genes that are classical markers of beta cells. And what was interesting, she found that, genes that were classical indicators of healthy beta cells, like PDX1, insulin, we found them to be highly expressed in the PFC slices. However, we saw a suppression of PDX1 intensity in slices that were on transwells. So the difference really is that PFC dishes, they allow for this superior sort of air-liquid interface where, oxygen can not only does it have to, can it like perfuse over the media into the tissue, but also underneath it. So, the slices essentially are much better oxygenated, in comparison to the transwell, which essentially leads to the choking of the slice or of the tissue. So we published this in Nature Communications, and the goal of actually doing this, like you may ask the question, well, okay, that's great, but a lot of studies can be done acutely on pancreatic slices. Well, as a developmental biology lab, we were interested, as sort of Juan very nicely outlined, looking at regeneration in the slice. So we needed a model. As a developmental biology lab, we needed a model where a tissue system could be alive or survive for extended periods, because we need to be able to treat tissue with a certain biological molecule. BMP7, in our case, is our molecule of interest. And then later look at events of transitioning events where non-endocrine cells would transition into endocrine cells or other cells of the pancreas, right? So in order to create this, in order to visualize this, we needed to create a model that would allow us to basically track a slice over time. And we showed some preliminary experiments, using this model in this paper. But what Mayur really did with Juan they said that, well, what if we would sequentially, in an orthogonal manner, take slices, dissociate them, and do single-cell RNA sequencing? So that's what he's going to talk about, his paper, which is up next, I believe.
Thank you, Fahd. So we'll come back to Miami here. Let me share the screen from here. And now All right, so I guess that you see that. So thank you so much for this introduction, Fahad, and I think that it's time for us to get into the paper, and Mayur will take over in a couple of minutes. Simply to reiterate what Fahad said, the point of doing this model is that now we have the means to look at the composition of slices at the single cell level over time in the presence or the absence of BMP7. So that's what we did using slices from the same donors, the same human donors. We got different data sets at time point 1, 5 days, time point 2, 10 days, with or without BMP7, and see what happens. So I'll let you take over from here,
Thank
Mayur.
you, Dr. Dominguez, Bindala, and Dr. Fahd. After this single cell RNA sequencing data analysis, first we receive the data and we perform analysis with the help of CRAT package, which developed by Satija lab. So CRAT algorithm allows the cells to group together in a cluster based on their similarity in transcriptomics. And as you can see over here, we identify the cell types of these clusters based on their highly expressing cluster markers. So you can see here we identify the clusters with ducts, acinar, and islets. If you see the presentation over here, how the CRAT organize the cells in the groups, and then also if, you can see the subpopulation of ducts, acinar, and then you can see the UMAP, that is uniform manifold approximation projection, that the cells are plotted in UMAP. So each dot here you can see over here represent a single cell, and we like always the analogy of continents and countries. So I can show here, this is like 18,000 single cells of particular time point of a control group, and you can see the ductal continent on left side, where various subpopulation subclusters are showing in the ductal countries, and on right hand side you can see the acinar continent, where clusters of acinar are shown as a represented as acinar countries. So we like to project this UMAP in cartographical maps with so we plotted this 2-dimensional UMAP into the cartographical map with the help of Bores package. So you can see this UMAP on a world of map, where you can see the acinar continent on left side and ductal continent on right side, and there are other islets, macrophages, endothelial, different clusters also.
Very creative.
Yeah. So then, as you can see here, this is a control data set for time point 1 that Dr. Bendala already showed in the previous slide. So you can see here the acinar and ductal continents are very far from each other. They are kind of separated. However, when we plotted the time point 1 BMP7 data set, you can see there is a nice bridge connecting acinar to ductal continent here. We got really interested with that. When we checked the gene expression of the cells in this particular disease cluster that we called it, these cells were expressing both acinar and ductal genes. So they were kind of hybrid clusters. So in order to dig deeper in the analysis, so we combined this control and BMP7 time point 1 data sets and made the combined UMAP and perform RNA velocity and trajectory analysis. So with the help of scVelo packages. So what it does is scVelo, it's like it identifies the RNA dynamic changes in gene expression and calculates the RNA velocity based on the unsplice and splice ratio of mRNA for each gene. And the PAGA package actually takes the information from the RNA velocity and draws the conclusion, like by showing the arrows from one transition from one cluster to another. I would like to show you in a better graph, where here you can see the cartographical map of control plus BMP7 combined UMAP at time point 1. Here you can see the ductal continent on left side, and on right side there is a acinar continent, and there is also islet, distinct islet cluster on right hand side. So what we observed, that when we treated this pancreatic slices with BMP7, the pancreatic, so the ductal progenitor cells, they proliferate and they start showing CNR plus ductal gene expression. So they become kind of hybrid. And when we remove the BMP7, these cells differentiate into various acinar subpopulation. And you can see, interestingly here, the red arrow directing from hybrid cluster to new islets that is being produced near the ASEAN continent. So we got really interested. Like there is already a pre-existing islet distinct populations cluster is already present, and the new islet is formed near the ASEAN continent. When we, as you can see here in 2D plot, they appear very far from each other. But if you can see on left side of the side of your screen, when we plotted this 2D UMAP and transform into 3D UMAP that is developed by Dr. Fahd, so you can see this uh, two islands. So we call this two islet cluster as islands of islets. So these two islands are kind of connected to each other. So red color cells, they denote, they represent the islets that is present on the right side of the UMAP, and the green color cells, they represent the islets that form near the ASEAN continent. So they are kind of connected to each other. We got really interested with this data, and we decided to dig deeper. So what we did is we subset this islet cluster from the whole UMAP and then perform recluster analysis with the CRAT. So you can see over here that after analysis, we observed three clusters of islets. So third cluster is being most mature, because the most of the cells, they were expressing betas, like insulin expressing genes, and this particular cluster harbors half enough BMP7 and untreated cell. Then-
Mayur, we had a question. I wanted to just throw it out there before you continue.
Yeah.
Very interesting work. I wonder, in your single cell RNA-seq, did you pick up any PSCs, pancreatic stellate cells, cluster over the time period? Did BMP treatment influence the cluster size?
Yeah, but that I can show you, like, but in cluster 2, but obviously BMP7 affect the cells, progenitor cells, to proliferate, and then after removal of BMP7 it differentiate, obviously. But I can talk this in like next this slide, like in 2 minutes. Like definitely the BMP7 affects the size of the cluster size, obviously.
Okay.
Yeah. So then this is a cluster 1 where we saw this is the most immature cluster that we observed, where most of the cells were expressing more ductal genes and less insulin gene expression. And this particular cluster also harbored by half and half BMP7 and untreated cells. But the most interesting part over here is the cluster 2. This clusters 2 is mainly 99% of the cells came from the BMP7 origin, and this cluster is kind of intermediate. It's in kind of transition state where it was showing the insulin expression in between cluster 1 and 3, and this particular cluster was showing more ASEAN genes. Even the RNA velocity also confirmed that this cluster was like in transition between cluster 1 and 3. So we would like to explain this in the context of like world of map, where when we treated this pancreatic slices with BMP7 treatment, this ductal progenitors proliferate and make more ductal cells, and they also make hybrid ducto-acinar cells. So after BMP7 removal, this acinar endocrine precursors, they actually either differentiate into acinar cells or make endocrine cells, new endocrine cells. And in case of like, if I would like to explain in the context of like islands of islets cluster analysis, where the most immature one, the cluster 1, is one where it shows more ductal gene and less insulin expression. The cluster 2 is like kind of acina endocrine precursor, where it shows more insulin expression compared to the cluster 1, but it's more acina origin. And the third one, that's the most mature one, where it shows a new endocrine cell as well as the pre-existing ilex, which shows more insulin expression. That's the story.
There's one more picture there. Right?
Yeah. So we would like to show this in future in case of spatial transcriptomics analysis like a nanostring geomics, and if there are pre-existing islets, or there is a new formation of beta cells can be formed or not. So I'll hand over to the. Thank you.
Wow, that's really, really interesting. Thank you so much.
All right, let's stop sharing
Yeah.
so that Isabella can share her screen.
Okay, let me know if you're able to see mine.
We can see, yes.
Yes.
Okay, perfect. Okay, thank you, Mayur, for your explanation. So basically, moving forward, one important aspect of the study was to validate all of this, complex bioinformatic analysis, and especially establish whether these newly created beta cells that arose from the BMP7 treatment are functional. So if they produce and secrete insulin in response to glucose stimulation. So to do so, we obtained human pancreatic slices from type 1 diabetes donors. We cultured them, as Fahd explained before, and we treated them, we infected them with some viruses. Basically, the viruses allowed us to tag different populations of cells within the slice. Basically, all of the cells that are non-beta cells are going to express a red color, and all of the beta cells, all of the cells that produce insulin, are going to express a blue color. This will allow us to visualize the transition when we treat our slice with BMP7, that it goes from a non-beta cell to a neogenic beta cell. And we usually see this purple transition-like stage until it fully transitions into a blue cell. Now, we also have an additional marker with these blue cells that it's going to kick in if insulin was secreted in response to glucose stimulation. So we do this to see the functionality of the newly created cells. Basically, this green marker, depending on the glucose concentration that it's in the media, it's going to express more green intensity. And that's represented here in this video. These blue cells are newly created cells from our treatment, and basically, as you can see right now, it's in 3 millimolar. Then we switch to 20 millimolar of glucose. That's a high concentration, and you see how the cells are responding. They're secreting insulin in response to high levels of glucose. And this pattern that we're seeing here, it's similar to that it's seen in a regular beta cell. KCl is also used as a positive control, and as you can see, the lighting of the cells is similar to the one we saw previously with the high levels of glucose. Here, this is showing how, well, with the slice, after we treated with the virus, you can see all of the cells. You can see that throughout the days with the treatment, there are no blue cells in the same area that we were imaging. And on day 7 and day 9, you can start seeing this transition like cells purple, and then eventually when you get to day 9, you get your beautiful blue cells that are newly created beta cells. We did the same type of experiment as that I showed before with glucose stimulation. So you can see that with high levels of glucose, the green intensity increases. That's also seen here in the graph. And these findings are remarkable because it's the first demonstration in a type 1 diabetic donor that you can regenerate functional beta cells with BMP7. Thank you.
Great. Those are the credits. Okay, so while they run, I'll just conclude by saying that beyond the interest in the basic science of regeneration, which is obviously a terrific approach, I think it is the first one where we can look at this in human-based models. There's most of the information that we have on regeneration is based on mice. it doesn't get any more real than a live chunk of the pancreas of a type 1D donor. And to show that we can regenerate those cells by adding a growth factor in the real thing, I think is extremely promising. What we want to do, obviously, at the end of the day, is to take this to the clinic. We want to use BMP mimetics that will induce beta cell regeneration in vivo without the need for transplantation. Obviously, that will have to be done at some point together with some immunotherapy to prevent the recurrence of autoimmunity, because otherwise we will exhaust the newly created beta cells or the pool of progenitor cells that will be destroyed again as they become beta cells. But there's a pipeline of clinical trials looking at this immune modulation, and we hope to be able to apply this strategy alongside. So that will be the subject of our next presentation. We are actively working on another paper in which we show that these BMP mimetics can cure diabetes in mice, and they actively induce regeneration in vivo. So that, together with the demonstration that it can happen as well in the slices from type 1D donors, I think it's great news for the field. In the meantime, as you very well know, the field is advancing very, very fast with the transplantation of human embryonic stem cells. And even though that may not be a functional cure for extended periods, let's say that, because one of the things that we haven't cracked yet is how to make a device that will contain those cells and preserve them from the attack of the immune system forever. But let's say that the cells that you put after transplantation may last for a few months, and then the patient has to come back for new cells every 6 months. That would be fantastic. It's not a cure, but it will be a life-changing thing for patients. But in the meantime, working on this. This is harnessing the real potential of the organ to replenish those beta cells. We know it happens. There's a lot of evidence, and now we think that we can accelerate the process tremendously by adding these BMP mimetics. So that's it. In summary, thank you very much to my other team members. I mean, I only presented the introduction, but they did all the work. So thank you very much for everything. And if you have any questions, we'll be here to take them.
I actually wanted to address the two questions, I believe, inside the chat. So
yes.
And Sarah, I had more of for a comment than a question, but he states that, very interesting. We tried transplanting human ductal acinar and islets to diabetic NOTS kids, but most acinar cells died after 12 weeks. So our model was the transplantation of these progenitor-like cells, right? In our PNAS paper, which was the transplantation of the ALK3 positive, P2RY1 positive progenitor-like cells. So we basically suspended them in a matrigel mixture and then transplanted them in the, the subscapular capsular space of the kidney, which is an ideal transplantation site, usually, very sort of frequently used for islet transplantation. So the total duration for our transplantation was about 4 weeks. So I'm not sure if that's one of the reasons. Another point I'd like to highlight is that we actually transplanted these progenitor cells, which transitioned into acinar cells. So the period during which they transitioned and they were acinar was probably around about, I would say, I'm guessing now, but it would be 1 week or 2 weeks. So definitely we didn't have the cells there for an extended period. We have no idea what happens to transplanted acinar cells over 12 weeks, I believe you're stating. So it's possible, in a situation where that acinar tissue is not draining its enzymes, I would expect that the pooling of the enzymes would cause the cells to die. So the adjustment of your experimental time period would be important, I would be assuming in that case. And the second question, I think Mayur was Shamik was interested in asking about what happens to pancreatic stellate cells. And I think this is a very interesting question because we've traditionally always focused on the endocrine or exocrine cell types, but we've never actually really studied stellate cells. And the stellate cells are always a part of our data sets. So we find these massive populations of CD90 or Thi1 positive stellate cells, both the activated and the quiescent sort of, pancreatic stellate cells. And of course, BMPs have an effect on them. And one of my conversations with Juan will always be is that, it could be possible that these stellate cells themselves are sources of BMP7 locally within the islet. It's just things that we haven't really ever investigated. But if you're interested, all the data is there, and Mayur, he's even uploaded an entire R Surat object to our data set on Geo. So anybody can easily download that and ask all of these questions, which is why we have all of our data sets open access for the community, for you guys to ask all of, these particular niche questions that you have regarding the data set. That's all. I don't know what the rest of the questions.
No, that's great. That's excellent. And everyone is very thankful for you addressing those questions. Can you, just one last question I had, can you, I may have missed this, but with the, can you detail or sort of like give high level details on the comparing the level of oxygen consumption in a ductal cell versus the beta cell? Obviously, the beta cell is very sensitive and needs high levels of oxygen consumption.
We haven't looked at ductal cells per se about their level of oxygen consumption. However, we do have data that we generated together with Chris Fraker, who is also here at the DRI.
We've had Chris on before. He's been great.
Yes. So we were differentiating embryonic stem cells all the way to beta cells, and we were measuring the oxygen consumption rate. And you can see that there's it goes up tremendously in, as the cells differentiate suddenly in the last stage when they become, from pancreatic progenitors to mature beta cells, there's a tremendous uptick of oxygen consumption. So clearly that was heading towards, the take-home message was that you cannot have a one-size-fits-all approach to culture cells, and you expect to differentiate your beta cells, with the same oxygen concentration at the beginning than at the end, because different cell types, as you progress, have different oxygen consumption requirements. We didn't see dogs in this, but you may argue that at the pancreatic progenitor stage, that was ductal-like, and it wasn't much compared to what you see after. Taking into consideration that these beta cells are among the most highly metabolically active in the body, so they require a lot of oxygen and energy. So that may be the reason why they also require a lot of oxygen.
Well, because I mean, it begs the question, if you can finesse the system so that you could basically, implant some sort of precursor ductal cells that are low oxygen consumers as part of an implant strategy, right? And then you drive them to become beta. So you've got a better chance of them, kind of acclimating as they get vascularized.
Exactly, and you said it. In fact, many of these stellate cells, if they go together in the implant, they would actually promote secrete VEGF and promote, neogenesis of new blood vessels, and that brings about obviously the increase in oxygen for the graft. But we are not in the business of engrafting the cells. We think that the pancreas can do the work itself, and that's the whole point. You have these stem cells or progenitor cells in the pancreas. Let's activate them using a chemical. It's not really a chemical. It's a growth factor that is natural. It cannot be any more natural than something that's made in the kidneys and circulates across the body. And one of the interesting things, and that's again for another discussion, is that these BMPs are not inducing proliferation left and right everywhere they go. It seems that nature or the body knows where they are needed. When we give these BMP mimetics to non-diabetic mice, there's no increase at all in the basal rate of proliferation in any organ that we have looked at. However, if you have diabetic animals, you see this only in the pancreas, because you need this injury, this insult to the tissue in order for BMP7 to actually act on those progenitor cells. So that's another advantage. You may not need to target this approach to the pancreas by giving it systemically. We think it's enough for it to reach a sufficient concentration in the organ and induce proliferation there and not anywhere else.
Wow. Well, this has really been exciting and excellent presentation, beautifully done, and really enjoyed the way you set it up. Just some great work coming out of the DRI at University of Miami, and I would encourage everyone to sort of keep an eye on this team, this sort of power team across different locations. really bringing some great work to bear. And we will host this on our website shortly as a recording, so those who missed it can also enjoy the presentation and reach out to you with any communications or collaborative ideas.
Thank you so much for having us, Monica. We look forward to collaborating with you in the future.
Same here. Have a great holiday season, and thank you again for all this excellent work you're doing.
All right. Thank you, Monica. Take
All
care.
right.
Thank you.
Bye,
Thank you
all.
so much.
Bye-bye.
Bye.
Bye.